Hello students, the topic of today's video is receptors. So in this video I am covering the cholinergic receptors, adrenergic receptors and G protein coupled receptors, and about them, their location, functions, selective agonist and antagonist, and how they can action. So let's start this video. First, cholinergic receptors. So cholinergic receptors are basically classified into two classes: muscarinic, represented by M, and nicotinic receptors, represented by N. So muscarinic receptors are G protein coupled receptors, and nicotinic receptors are ligand gated cation channels. These muscarinic receptors are subdivided into these subgroups M1, M2, M3, M4 and M5, and nicotinic receptors are subcategorized into NM and NN. So first we are talking about M1. So this M1 is present in a ganglia, means autonomic ganglia. Ganglia is the region where preganglionic neuron and a postganglionic neuron communicate with each other. So here, as you can see in this diagram, in both parasympathetic and sympathetic system, this preganglionic neuron receives signals from CNS and reaches at ganglia. Here it releases acetylcholine, and this acetylcholine binds with the M1 receptor and produces a signal for the postganglionic neuron, which ultimately leads to the message to the affected cell or organ. So when acetylcholine binds with the M1 receptor, it causes depolarization for the generation of new signal. So in autonomic ganglia, this M1 receptor stimulation results in a depolarization. And this M1 receptor is also present in a different parts of brain, mainly in the central neurons on cortex, hippocampus and corpus striatum. As we know that these three components are present in a forebrain region, which is responsible for the learning, memory and motor functions. So M1 receptor stimulation will result in a learning, memory and motor functions. This receptor is also present in a gastric gland, in a parietal cells and in a enteroendocrine cells. As you can see, this is the image of a gastric gland. Here in a parietal cells, this M1 receptor stimulation is responsible for gastric acid release, and in an enteroendocrine cell it is responsible for histamine release. Now coming to the M2 receptor, so this receptor mainly present in a heart along with a beta 1 receptor. So it mainly controls the heart activity by decreasing the activity. Okay, in SA node it causes hyperpolarization, which results in a decrease in rate of impulse generation. In AV node it decreases the velocity of conduction. In atrium, M2 stimulation is responsible for shortening of action potential duration, which ultimately leads to the contraction decrease. And in ventricle also it decreases contraction. M2 receptor also present in other parts of brain and is responsible for the tremor and analgesia. In cholinergic nerve endings, it decreases acetylcholine release, and in the visceral smooth muscles, it will show contraction. M3 receptors. These are present in eye. In eye region, in iris, contraction of pupil takes place, and it also causes ciliary muscle contraction. It is also present in blood capillaries. After its stimulation, it releases nitric oxide, which causes vasodilation. It also presents in visceral smooth muscles along with M2, and it also shows contraction. In exocrine glands, it will result in secretion. Now we are talking about the nature of receptor, its selective agonists and antagonists. So M1 having a GQ protein coupled receptor, means it's a GPCR having a GQ coupled protein, and it works by IP3 DEC pathway, in which it increases calcium in a cytosol, and it also activates a phospholipase A2, which leads to the prostaglandin synthesis. And M2, it has a GI or GO protein coupled receptors, means it's a GPCR having a GI coupling unit, and which directly affect on the potassium and the calcium cells without activating CAMP or without generating IP3 DEC, and it also decreases CAMP. In M3, it is same as M1 having a GQ protein coupled receptors, and it functions through IP3 DEC and a PLA2 pathway. So agonist for M1 is oxotermorine, for M2 is methacholine, and for M3 is bethanacol. And antagonist for M1 is pyrenzepine and talenzepine. For M2 is methoxytramine and tripentramine. M3, it is a darifenacin and a solifenacin. Now we are talking about nicotinic receptors. Uh, Here first I am talking about its location and function. So NM, as the name indicates, M means a muscular junction. So NM receptor is mainly present in a neuromuscular junction, and its stimulation is responsible for depolarization of muscle end plate and contraction of skeletal muscles. This now NN receptor is present in autonomic ganglia along with the M1 receptor. So in autonomic ganglia of both sympathetic and a parasympathetic system, its depolarization happens in a ganglia. Depolarization of postganglionic impulse. And NN receptor is also present in adrenal medulla. Its stimulation causes catecholamine synthesis, and in CNS it has a site-specific excitation and inhibition functions. Now nature of receptor. So nicotinic receptors are basically ligand gated cation channels. So NM receptor has an intrinsic ion channel. It is a pentamer of alpha, beta, gamma, epsilon and delta subunits. Each subunits have 4 transmembrane segments, and it is responsible for opening of sodium and potassium channels. And NN receptors, it is also intrinsic ion channel having a pentamer of only alpha or alpha beta subunits. It also has 4 transmembrane segments, and it is responsible for the opening of a sodium, potassium and a calcium channels. Now agonist for NM is PTMA, phenyl trimethyl ammonium and nicotine. For NN it is DMPP, dimethyl phenyl piperazinium and nicotine. Antagonist for NM receptors are tubocurarine and alpha bungarotoxin, and for NN it is hexamethonium and a trimethaphan. Now coming to the adrenergic receptors. Adrenergic receptors are also classified into 2 groups, alpha and beta. These both alpha and beta receptors are G protein coupled receptors. So alpha receptors are subdivided into 2 subtypes, alpha 1 and alpha 2, and beta having subtypes beta 1, beta 2 and beta 3. So about alpha 1, its location and functions. So alpha 1 is present in eyes, and it causes a contraction of radial muscles of iris, which leads to the mydriasis, and it also decreases the aqueous secretion. In intestine, it shows intestinal contraction. In urinary bladder, it shows detrusor muscle contraction. In heart, it has a little action, but when the alpha 1 drugs are given at a high doses, then it will shows arrhythmia. And it also shows a uterus contraction On blood vessels, it shows a vasoconstriction, which results in an increase in blood pressure. Here, alpha 1 and alpha 2, both receptors shows vasoconstriction. In liver, it shows glycogenesis, and in spleen, it shows splenic capsule contraction. Other than this, in a salivary glands, it is responsible for the secretion of water and potassium, and other gland secretion also happens, and it is responsible for gut relaxation. Now alpha 2. So alpha 2 is present in a pancreatic beta cells, which functions to decrease insulin release. Alpha 2 receptor also present in a platelets, so it is responsible for platelet aggregation, and as I already told you, it shows a vasoconstriction, and in brain it is present in a prejunctional on nerve endings at alpha 2A. So it functions are inhibition of transmitter release, and it also present postjunctional in brain, which decreases the central sympathetic flow. Now coming to the agonist of alpha 1 receptors, so these are phenylephrine and methoxamine, and alpha 2 agonists are clonidine. Antagonist for alpha 1 is prazosin and terazosin, and alpha 2 it is yohimbine. Nature of receptor. Alpha 1 is a GQ protein coupled receptor. It works by IP3 DEC pathway, so it increases cytosolic calcium, and it also activates PLA2, which increases PG synthesis. And alpha 2 receptors having a GI-GO protein coupled receptors, and it decreases CMP, opens potassium channels, and as well as calcium channels. Now we are discussing about the location and effects of beta receptors. So beta receptors are basically known as the cardiac receptors, because beta 1 receptors are responsible for the activity of heart functioning. And here, as you can see, in a SA node it increases the rate of impulse generation, in AV node it increases velocity of conduction, in atrium it increases contractility, and in ventricle it increases contractility. So ultimately it increases the functioning of heart. And a beta 1 receptor also present in the juxtaglomerular cells, and these cells are present in a juxtaglomerular apparatus, which is present in the nephrons of kidneys, and it is responsible for the release of renin. And beta 3 receptors are present in a adipose tissue, and it is responsible for the fat lipolysis, which ultimately leads to calorigenesis. Now beta 2 receptor, and beta 2 receptor is present in liver. So here in a liver it causes a glycogenolysis, which ultimately leads to hyperglycemia. It also causes augmented insulin and glucagon secretion, and it causes splenic capsule relaxation. In muscles, in an active state, it prolonged the fast contracting muscle and abbreviated in a slow contracting muscle, and also leads tremor. It causes uterus relaxation, vasodilation, bronchoconstriction, detrusal relaxation, and intestinal relaxation. The agonists of beta 1 receptor is dobutamine, and beta 2 is salbutamol and terbutaline. The antagonists for beta 1 are metoprolol and atenolol, and beta 2 are alpha methylpropanolol. Now let us talking about G protein coupled receptor. As we already see that all the muscarinic receptors, alpha receptors and beta receptors are consist of these G protein coupled receptors, GPCRs, working through different mechanisms. So let us discuss these different mechanisms here. So in this chart, I hope you can it is visible for you all. So in this chart, these in on a cell membrane, sorry, so in a cell membrane this is blue, pink, red and green, and these are G protein coupled receptors. These are GPCRs which are working through different mechanisms. So first one, it is a GS GPCR. Its coupling protein is a GS, and here when a ligand binds at a ligand binding site on a G protein, so it changes the conformational structure of the protein, which ultimately leads to the activation of alpha subunit, and alpha subunit is activated by the attachment of GTP. So after GTP attachment on alpha site, so it detached itself from beta and gamma subunits, and this GTP containing alpha subunit uh, goes to activate adenylyl cyclase. It is present in a membrane, and this activated adenylyl cyclase converts ATP to cyclic AMP. Cyclic AMP here is the second messenger, and this cyclic AMP activates protein kinase A. As the name indicates, kinases are those enzymes which are responsible for phosphorylation. So this pKa increases protein phosphorylation and also opens the calcium channels in a myocardium and skeletal muscles. And the receptors comes in this category are beta adrenergic and prostanoid receptors. And second one is GQ. It means its coupling protein is a GQ. So here when a ligand binds with the ligand binding site, it changes its conformational structure, which leads to the activation of alpha subunit, and this alpha subunit along with GTP detached itself from beta gamma subunit and activates phospholipase C, PLC, and this PLC activated PLC again split or break down this PIP2, means phosphoinositol bisphosphate, into two units, IP3 and DAG, inositol trisphosphate and diacylglycerol, and this IP3 leads to the activation of PKC, phosphokinase C, and this DAG molecule increases calcium release. So these two pathways shows increased protein phosphorylation and also activate calcium binding proteins. And the receptors which are working through this mechanism are muscarinic M1, M3, alpha 1 adrenergic, serotonin 5-HT1 and prostanoid. Third one is the GI, means inhibitory GPCR. Here after the activation of alpha subunit, there is a GTP containing alpha subunit detached itself, but instead of activating adenylyl cyclase, it inhibits adenylyl cyclase, so that inactivation or inhibition of adenylyl cyclase will leads to the inhibition of further steps. So the receptors working through this are muscarinic M2, dopamine D2, alpha 2 adrenergic, GABA B, serotonin 5-HT1 and prostanoid. And last one is the GIGO. Actually in this it is the step where instead of alpha subunit, this beta and gamma subunits work here to activate the potassium channels as well as other calcium channels. So here this beta gamma subunits activate or responsible for the opening of potassium channels, so that exit of potassium takes place. So in this way it causes opening of potassium channels in heart and smooth muscles, as well as inhibit neuronal calcium channels. And in this way for the calcium increase, adrenergic beta 1 receptor is responsible. For calcium decrease, dopamine D2, and potassium increase by alpha 2 adrenergic, muscarinic M2, dopamine D2 and serotonin 5-HT1A. I hope you all understand this chart, this flow chart, and if you are having any kind of doubts, so you can comment me in our comment section, and I hope this video is useful for you guys. So If you really like it and you get a knowledge from this video, then you can like this video, share this video, and subscribe this video, subscribe my channel for more upcoming videos. And thank you.